So, what is essential dogma of biology? Well, if you take a look at the cell here, this is a eukaryotic cell. It doesn't matter what cell that is, but we know it's a eukaryotic cell because you have a nucleus here, you have a nucleolus right here, and you have the plasma membrane of that cell. So the central dogma of biology, I'll go ahead and label this stuff. This is just the nucleolus. This is the nucleus, and this is your lipid bilayer membrane, also called the plasma membrane. So the central dogma of biology is basically this flow of information that goes as following. DNA makes RNA, makes protein. Now the process of going from DNA to RNA is transcription, and the process of going from RNA to protein is translation. And so we begin with DNA. And where do you find DNA? Well, you find DNA right here in the nucleus. And so in humans, for instance, you'll find DNA as a bunch of chromosomes, and you have 46. And so if you need to create a particular protein, let's say some sort of an enzyme, what you have is you have a gene within one of these chromosomes that's going to code for that protein. And a gene is made up of a bunch of DNA. And so let's say that gene is within that chromosome right here, within this sister chromatid of the previous chromosome. So you have another sister chromatid. Let's say you have a gene right there that you want to transcribe and translate. What's going to happen is this gene is going to be transcribed into what we call messenger RNA. And actually, what's going to happen first is you're going to make what's called heterogeneous nuclear RNA. And then this heterogeneous nuclear RNA is going to be processed into messenger RNA. And so this product, messenger RNA, is what you get after transcription. Once you have that messenger RNA, the messenger RNA is going to leave through nuclear pores, these small sort of pores within the nuclear membrane. And so the messenger RNA will leave, and it's now in the cytosol of the cell. And once in the cytosol of the cell, this messenger RNA is going to be translated into a amino acid sequence. And this amino acid sequence is going to be a polypeptide that's made up of, generally speaking, hundreds to thousands of amino acids. This is the process of translation. Now, once you have these amino acids are going to go through a process that's called protein folding, and you're going to end up with a protein product that is 3D in orientation and shape. And so you might make an enzyme that looks something like this. And this would be your final protein product. So the idea of the central dogma of biology is to grab the genetic information within our DNA, right here, this chromosome. We grab genetic information from it. We took a particular gene and transcribed it into messenger RNA. First, we made, of course, the pre-messenger RNA. We call it heterogeneous nuclear RNA. We processed it into messenger RNA. Processing RNA is for another video. We grabbed the processed messenger RNA. We took it outside the nucleus. We translated it using ribosomes into, again, that's for another video, but we translate it into an amino acid sequence. And then that amino acid sequence went through protein folding to create this functional product. Thank you for watching.